Hello everyone, I'm Qianwen Wang, currently a postdoc at Harvard Medical School. Today I'm presenting our study, Interactive Exploration of Tissues and Cells Guided by Explainable Visual Pattern Mining. To start with, let's talk about the visual exploration of small multiples. One commonly used method is to present those items using latent vectors that are learned by machine learning models, and then display all those items in a scatter plot by applying direction reduction method to the latent vectors. While similar items can be identified in this method, it suffers from a lack of interoperability. For example, it has a vague definition of what are similar items. It is possible that the users are only interested in certain visual patterns and want to leave others when they group similar items. In our previous study, we applied disentangled representation learning to address this issue. This method allows us to generate latent vectors that disentangle visual patterns and encode them as separated dimensions. For example, here, the value of one dimension indicates whether a person is smiling or not, and by gradually changing the value of this dimension, the facial expression changes with it. Also, since it is an unsupervised learning method, those latent vectors can be learned without the tedious human labeling. We further developed an interactive visualization tool to support the visual exploration with those explainable latent vectors. This tool allows us to arrange, group, and summarize items using those latent vectors that are explainable. In those posters, we report how we apply this visualization tool to the analysis of tissues and cells through two use cases. In the first use case, each item is a cell that is segmented from the single-cell images. We identify the boundaries of cell and the nucleus from those single-cell images. In the left side concept view, each row is a latent dimension. For example, the value of this dimension is related to the cell size, and the value of that dimension is related to the cell orientation. Users can also rename a dimension as needed to facilitate the analysis. In the right side item browser, users can use the visual concept that they identified in the concept view to assist the visual exploration of all items. For example, here, while the UMAP projection did not reveal clear insights about those items, we can group those items based on the visual patterns that we identified in the concept view. We can investigate the correlation between cell size and the other item labels that are imported from the metadata. For example, here we can see the larger cells on the left side tend to have different item labels than the small cells on the right side. We can also examine the item distribution based on the sizes and orientations. Here we can see that the small cells and the larger cells tend to have different distribution in the orientation. In the second use case, each item is a patch extracted from stained host-net images of breast cancer specimens. Again, we start with the default UMAP projection, where some similar items are revealed. Those projections is not easy to interpret. Therefore, we organize all those items using two concepts we identified from the concept view. One is related to the tissue density, and another is related to the color of the stained tissues. By adding labels that indicate the cancer cell presence to each item, we observe a strong correlation between the presence of cancer cells and the two visual patterns. We first analyze items that had confident false negative predictions. We import labels from the metadata and using them to filter items. We can see that almost all those items are loose purple tissues. From the context view, we observe that those items are close to each other and are more likely fat tissues that are surrounded by cancer cells. Such an observation can help us understand which types of items are hard to classify, therefore guiding the diagnosis and refinement of the classifier. That's all. Thank you so much for listening. You can find more information at our preprint, and you are welcome to contact us for questions and discussion.